This is your instructor Kwame Shen, and for this lecture, we are looking at prokaryotic cells versus eukaryotic cells, and I've named this the anatomy of cells. It's going to be a two-part lecture, so that's the first part of two lectures. In totality, at the end of this lecture, the two-part, that we would appreciate plasma membranes better and understand the complexities of it, how transportation generally happens between cells and their environment, look at prokaryotic and eukaryotic cells in terms of the common structures that they do have and the differences that exist between these groups of cells. Cells generally, which are the basic building blocks of our lives, are made up of atoms that have combined to form elements that have come together to form molecules, and then macromolecules such as proteins, lipids, and nucleic acids. Cells provide structure, are able to define the contents that they enclose, are able to carry out certain metabolic activities. They contain specialized nucleic acids, DNA, and they're able to replicate this DNA within themselves. So there are basically two groups of cells, the prokaryotes and the eukaryotes. They all have a cytoplasm and a plasma membrane. So that's one commonality that you find between these two types of cells. There are more differences between the two, and we'll delve more into that. But first, we'll look at the cell membrane or the plasma membrane. The cell membrane is made up of phospholipids bilayer. A phospholipid is composed of phosphates, and glycerol, and fatty acids. The fatty acid component is described as hydrophobic because it doesn't easily interact with polar substances such as water, because it's oil and water don't really mix that well. And the head is made up of the phosphates and the glycerol, and these are more water soluble, so they are hydrophilic. So there is a hydrophilic head and a hydrophobic tail, and whenever the cell membrane comes in contact with water, it rearranges itself to form this bilayer, where the tails are towards each other and the heads are pointing outward and interacting with the water that is outside or inside the cell. And the cell membrane serves several roles and very important functions for the cell. It defines the cell, so we're able to tell what's inside the cell from what is outside the cell. It allows certain substances to pass through, and so substances may pass into the cell and become raw materials for cellular activities, or certain waste substances that are no more needed may be moved out of the cell. And it's all because of the cell membrane that these activities take place. But what about molecules that cannot go across these membrane for one reason or the other? And that is where we see the role of specialized proteins that are embedded in the plasma membrane. And these proteins may be peripheral proteins that are just on the outside, on the surface of the cell membrane, or they may be integral proteins that are found embedded into the cell membrane or plasma membrane, then they actually are found to be all the way across, so from the outside to the inside, and they play very critical roles. So these embedded proteins will help in transportation. They help in cell recognition. Cells have these glycoproteins on their surfaces that allows them to recognize each other. So a cell can recognize neighboring cell because they have this lock and key arrangement that when it fits into, they can actually tell that, oh, this is a neighboring cell and that is another cell, and they are all part of this one organism which we are part of. And cells, the cell membrane also have these features that also help us to sense, be able to read its environment. So it can tell if there is a change in the environment and certain measures that it has to take. And usually these changes may actually begin with some of these molecules entering the cell, and then the cell becomes aware of the environment. Cell membranes or plasma membranes are not static. They are not just fixed. They are very fluid. So we have what we call the fluid mosaic model, which explains the nature of plasma membrane. And it's important that you understand how this works. The phospholipids and the proteins in the membrane are frequently moving. Okay? So they move freely within the membrane. So for instance, you may have lateral movement of phospholipids. So this may move to the side and that will move to that side. So there's this movement going on. You could also have a flip-flop where it turns on its head, and so this becomes this, and that changes position with that one. So these movements are always going on within the fluid mosaic model. The importance of such a model is that it makes it easier for cells to fuse. So cells can fuse, cell membrane can easily fuse with vesicles, which are also made up of similar membranes surrounding them. It's easier for cells to attach to one another. It's easier for other molecules to attach to cells and be able to enter the molecules that are needed. It makes it easy for substances to move within the membrane because it's not static, it's always moving. Allows membranes to proteins to move also within the membrane as needed. And one more important factor, which we especially see in prokaryotes, is that because of the fluid mosaic nature of the membrane, during cell division, after the nucleic acid has replicated, and we have equal copies of it, the cell membrane or plasma membrane also has to divide equally. The fluid mosaic model is able to explain how the cell membrane is able to divide equally or evenly between the offsprings. The fluidity of it also means that when temperature is very high or very low, it still is able to maintain some functionality as a membrane, as a protection around the contents, unless the temperatures go beyond certain ranges. So it gives it that kind of a buffer protection in terms of temperature changes. And you also have cholesterol being an important component of the fluid mosaic model. And the cholesterol, what cholesterol does is that it prevents the membrane from hardening. So you have this bilayer that is very fluid, a lot of movements going on, because within them there are both saturated and unsaturated fats, and they have to be combined in the right proportion. If you have only fatty acids that are saturated, being part of the phospholipid bilayer, what will happen is that if you arrange them, they all get stuck together so tightly. If you only have unsaturated, If you stick them together, there'll be too many spaces. So it's a combination of the two. And what happens is that because of cholesterol, the presence of cholesterol in there, which will have a mixture of both saturated and unsaturated fats, the cell membrane or plasma membrane isn't rigid in the presence of cold. If there was no cholesterol, it would become very rigid. If there was no cholesterol, there wouldn't be any flexibility, and then it will end up becoming very brittle and end up breaking. In very hot temperatures, in a very hot environment, without cholesterol, the cell membrane or plasma membrane will become too fluid or too flexible, and it wouldn't hold. Anything can just pass through because they'll be too apart from each other. And so these are very important points that you need to note about the importance of the fluid mosaic model. The peripheral proteins, which I've already alluded to, lies on the outer surface. So these are peripheral proteins. And then you have integral proteins that are distributed within the membrane. The cell membrane regulates, one of its functions is that it regulates movement of substances across into the cell and outside the cell. And so it is the means by which the cell is able to have access to nutrients, be able to get a supply of oxygen that would use in metabolism, be able to maintain the right pH, and secrete waste outside to the outside environment, and be able to synthesize enzymes and release them into the environment when needed. So how does this movement of substances into and out of the cell happens? We have what we call passive movement of substances, and then we have active movement of substances. The passive movement of substances does not involve the use of energy. So we have simple diffusion and facilitated diffusion. A simple diffusion is when you have movement of substances in the direction of the gradient. So for instance, if we look at two concentrations, there is a membrane here, you have concentration 1 and concentration 2. What happens in simple diffusion is that where you have a high solute concentration, these solutes will move across the membrane to this other chamber where there is less or fewer solutes until an equilibrium is established. Now, when an equilibrium is established, it doesn't necessarily mean there are no movement across the membrane. However, the movement across the membrane is such that at the end, the net flow is not in either direction. There is a net flow of 0 across the membranes because they cancel out each other. And simple diffusion will happen until there is equilibrium. Simple diffusion doesn't require a membrane. And so, for instance, if you have high oxygen concentration outside the cell membrane, and the cell needs this oxygen, all that it does, it's going to diffuse across the plasma membrane from where there's high concentration to where the inside where there is a lower concentration. So the simple diffusion taking place. Then there is facilitated diffusion. And for facilitated diffusion, there is some help. It is facilitated. However, there is still no use of energy. So there is the use of what we call transporters. And these are membrane proteins, integral proteins that are used for the transportation of these molecules from areas where they are in high concentration to areas where they are in low concentration. So you have two different types of these transporters. You have the non-specific, where the molecules that go through doesn't have to have a specific shape or form. Okay. So far as they're in high concentration and the membranes are available, they're able to use them to, once it touches the opening, it opens up and it's able to pass through to the other side. And then you have specific transporters. And for specific transporters like the lock and a key, the shape of the molecule that is trying to enter the cell has to have a specific characteristic. Now, once it has that specific characteristic and it fits into the, in this case, the opening in the protein, the integral protein, it just opens up and it's able to come to the other side as well. So glucose is transported, for instance, using this method. Note that energy is not required in facilitated diffusion. Then there's osmosis, which is the diffusion of water molecules from a region of high water concentration to a region of low water concentration. So this happens when solutes are not moving or cannot move, and so water does the movement. So it's a diffusion of water. So water is in high concentration here in this chamber, water is in lower concentration here in this chamber. So water will move from this region of high concentration to this chamber of low concentration until equilibrium is established. And energy is not expended in that process as well. So a cell may behave in a number of ways based on the environment that it's placed in. So if you have a cell that is placed in an environment where the concentration of solutes outside the cell is the same as the concentration of solutes inside the cell, then the net movement of water molecules outside or into the cell is going to be zero. So there is no net flux in terms of movement of water. And such an environment is described as isotonic. So it's the same conditions inside the cell as pertains outside the cell as well. A cell, however, could also be placed in a solution where there are fewer solutes. So there is more water outside the cell than inside the cell. Osmosis is going to happen, and when osmosis takes place, there's going to be movement of water outside the cell into the cell until it gets to the point where the cell bursts. Then there's hypertonic solution, where a cell is placed in an environment where there's more solutes outside the cell than inside the cell. There's more water inside the cell than outside. So water molecules will move from a region of high water concentration to a region of low water concentration. And that will result in the cell shrinking because it loses water, becoming what we call plasmosized. So we have isotonic solutions, we have hypotonic solution where the cells will swell. So we are using red blood cells now. And then you have hypertonic solutions where the cell loses water and shrinks or shrivels. Okay. So in addition to the passive movement of molecules across membranes, there is the active movement of molecules across membrane. Membranes are such that in addition to the integral proteins helping molecules move from an area where they are in high concentration to an area where there is low concentration, facilitated diffusion, there are instances where you have more of the molecules, let's say, within the cell, but the cell needs even more of it. So in that case, you're not going with the concentration gradient, you're going against the concentration gradients, because there is more inside the cell than outside the cell, but the cell still needs more of it. Now in this case, you need energy to move these substances into the cell, because you're moving it across against the concentration gradient. And that is where active transport comes in. So energy is expended in moving these molecules across the membrane, and they may be moved either through special proteins, integral proteins, or even through the membrane itself, plasma membrane, directly. So it allows for the transport from outside to inside despite or in spite of their concentration gradients, so that the cell is able to accumulate the amount of raw materials that it needs for its processes. There is what we call group translocation, and group translocation occurs exclusively in prokaryotes. So it's a type of active transport that occurs only in prokaryotes. And what happens is, it's very interesting that molecules are moved against concentration gradients, but however, the mechanism is such that when the molecule moves across the membrane, it's reconfigured. The molecule changes, it's altered, and once it's altered, it cannot go back through the channel that it came through anymore. So that's how these microorganisms are able to move substances against concentration gradients and then keep them within the cell and be able to use them for their cellular activities. So by way of overview, we have passive transport, where there is movement of molecules in line with the concentration gradients from high concentration to low concentration, and energy is not used. Integral proteins may be used, either non-specific proteins, protein transporters, or specific protein transporters may be employed. Then you have active transport, which involves the movement of molecules against the concentration gradient, but the most important factor here is that energy is expended. So moving on to the cells that we have, the two groups of cells. So we have prokaryotes and eukaryotes. Prokaryotes are characterized by a number of features, and so we'll look at some of them. You have the Glycocalyx, which is a type of polymer, very jelly-like in nature, that is secreted to the outer of these microorganisms, or prokaryotes. And so it's secreted to the external of the cell wall. If they are well organized and firmly attached to each other, closely packed, it becomes what we call a capsule. So you have certain bacteria that have capsule, like Klebsiella pneumoniae has capsules. If it's not well organized and it's quite loose around the cell wall, it is called a slime layer. So this is a slide of a bacteria with capsules, and the capsules are those spaces that you see around the bacteria. Prokaryotes may also have flagella. The flagella are long hair-like structures that are found on the bacteria, and it helps them to move. There are some bacteria that do not have any flagella. Those are called atrichos. So these are atrichos bacteria or prokaryotes. There are those that have several flagella all over their bodies. Those are the peritrichos. And then there are those that have they have these flagella on either one side, one pole, or on both poles of their cell structure. And these are polar. These are described as having polar flagella. They may have just one. If it's just one, it's a monotrichos. So this is a monotrichos. You may have several of them coming from just one pole. That is it's still polar, but it's lophotrichos. It may have two, each coming from one pole, and that is amphitrichos. So An example of a bacteria that uses motility quite well because of the many flagella that it has, and those flagella are mainly peritrichous, are bacteria that belong to the genus Proteus, the Proteus. And so this is an example of a bacteria that has been incubated. It was incubated at a center like that, and after a period of incubation, we see that it has moved all over the plate in this wave-like fashion. And so Proteus vulgaris, Proteus mirabilis are examples of bacteria that do that. I have a short video showing the movement of bacteria in here. So you see the very sharp movements that are taking place, and then you see this twitching movements, more of a tumbling that is happening. And it all comes down to how the flagella is rotated. Is it rotated clockwise or counterclockwise? If it's rotated in a clockwise direction, it results in a tumbling. The flagella is rotated in a clockwise direction, results in a tumbling. If it's rotated in a counterclockwise direction, it results in a running or a taxiing. So the taxis is a movement of bacteria towards or away a stimuli. So when the bacteria is moving towards a stimuli, it moves with a certain purpose. If it's moving towards a chemical, it is described as a chemotaxis. Towards light, and that becomes a phototaxis. Prokaryotes or prokaryotic cells also have filaments. They have these structures that are found on them, and for instance, we have what we call axial filaments. The flagella that are under an outer sheet, and they spiral around the cell, and we find these in the spirochetes bacteria. So this is an example of an axial filament. And so you have these structures that are going around or running around the bacteria. Then you have pili. Pili are short hair-like appendages. You also have fimbriae. Pili are a bit longer than fimbriae. The pili helps the bacteria to move. So two types of movement, the twitching and the gliding movement. Both pili and fimbriae are made up of protein called pilin, Pilin, and we see twitching motility in bacteria such as Pseudomonas aeruginosa and Neisseria gonorrhoeae. We also see gliding movement, also caused by the pili in myxobacteria. The fimbriae are shorter appendages. The fimbriae are involved in the formation of bioforms, help bacteria to attach to each other and to surfaces, which makes them more resilient to treatments and increases their survival in even harsh environments. Then we have cell walls. The cell wall is semi-rigid. Note that the cell wall and the plasma membrane are not the same. The cell wall is made up of a network of macromolecules called peptidoglycan. And the peptidoglycans are examples of polysaccharides, which are polymers. And these polysaccharides are intertwined with peptides. So you have cross linkages of polysaccharides and peptides. The main function of a cell wall is to give the cell a lot more resilience, a lot more integrity, so that when the cell is placed in a solution with fewer solutes than the cell contains, the cell doesn't rupture. It also maintains the shape of the bacteria and helps the flagella and other surface structures to be able to be embedded into the cell of the bacteria. In terms of pathogenicity, it contributes to how pathogenic a particular bacteria may be. So certain bacteria are very pathogenic, mainly because of the cell wall that they have, because it gives them some resilience to treatment, and so it makes them more pathogenic. They could also, however, be the site of antibiotic action. So, for instance, we have penicillin, which prevents the formation of peptidoglycan. So gram-positive bacteria, which has a huge component of peptidoglycan in their cell walls, are not able to survive in the presence of penicillin. Well, until not too long ago, when they started building resistance. But the reason why they were effective, because they prevent or they interfere with the formation of peptidoglycan in the cell walls of bacteria. So over here we see a demonstration, a picture of peptidoglycan. These are the monomers, and so the monomers, you have the NAM and the NAG. The NAM is N-acetylmuramic acid. The NAG is N-acetylglucosamine. So N-acetylmuramic acid and acetylglucosamine. And they form an alternating, like they alternate with each other when they are forming the beta-1,4 glycosylic bonds. They also have cross linkages. So there are cross linkages with amino acids that form peptide bonds. And there is also another linkage that they form. So there is a cross linkage. There's a linkage here and there's a linkage here, and then it joins the two together. So they all join together like that. So gram-positive and gram-negative cell walls are different. They're unique. And the uniqueness comes down to the content of peptidoglycan. So this is the cell wall. These are peptidoglycan. You see the alternating NAM and NAG. Okay. You have a similar one here, but look at the contents. The amount of peptidoglycan that you find here is really small compared to the amount of peptidoglycan that you find here. Okay. And it confers different properties on them. So gram-positive bacteria have many layers of peptidoglycans, giving it more rigidity, thickness in its wall. Gram-negative bacteria do not have these extra layers. They only have a very thin layer of peptidoglycan. However, they have an extra outer layer. So in gram-positive, you have a plasma membrane, and then you have a thick wall of peptidoglycan interspersed with lipoteichoic acids, all giving it some integrity. However, if you look at the gram-negative bacteria, they are very thin peptidoglycan, but they have an outer membrane. So in addition to the plasma membrane, they have an outer membrane. So these are some of the differences that you can see in the cell walls of gram-positive and gram-negative bacteria. We used our understanding and our knowledge of the cell wall of these bacteria to actually separate them. So we have gram staining, and we're able to differentiate gram-positive from gram-negative bacteria by using dyes. So you have crystal violet dye, iodine is added, and so there is a chain in terms of a complex formed between the crystals, the crystal violet compounds, and iodine. If the compound, if the organism is gram-positive, has a lot of peptidoglycan in the cell wall, it will retain this color, the crystal violet color, so that even if you wash it with alcohol and counter stain it, it will still retain the crystal violet color. And those are the gram-positive bacteria. And the bacteria that will lose the crystal violet color and then pick up the counter stain, which is safranin, are then described as gram-negative because they have a thinner peptidoglycan in their cell wall. You have the cytoplasm. The cytoplasm of prokaryotes contains about 80% of water that is mixed with enzymes and lipids, so it's pretty thick. In the cytoplasm, we find a cytoskeleton, a nucleoid, ribosomes, and endospores. So I'm going to end the first part of the prokaryote versus eukaryotes here, and the next lecture will begin again at cytoplasm and continue from there.